Okay, you guys, this is the first section of Chapter 20. So we just finished up Chapter 18, which is about viruses and bacteria, which the purpose of that was to introduce you to some things about viruses and bacteria, and then we're going to build on that in Chapter 20, which you can see the title says DNA Technology and Genomics. So the purpose of this is we're going to use some of the things that we learned about with bacteria and viruses and apply that to manipulating them to do some things with DNA based upon what we've learned with bacteria and viruses. All right, so the overview for the whole chapter. So I'll just read through this and discuss it. So we're going to talk about sequencing of the human genome, largely completed by 2003. DNA sequencing has depended on advances in technology, starting with making recombinant DNA. So we're going to talk about that. In recombinant DNA, nucleotide sequences from two different sources, often two species, are combined in vitro, that means in the laboratory, into the same DNA molecule. Methods of making recombinant DNA are central to genetic engineering, the direct manipulation of genes for practical purposes. So we're going to talk about how we manipulate genes to do some things for us. And then DNA technology has revolutionized biotechnology, the manipulation of organisms or their genetic components to make useful products. And an example of this type of technology is something called the microarray, the measurement of gene expression of thousands of different genes. And so we're going to talk about all of those kinds of things. So they're real-world things that we do as a result of knowing some of the things that we know. So this first section says, the title is DNA cloning permits production of multiple copies of a specific gene or DNA segment. So we're going to talk about how we clone and take one segment of DNA that codes for making a gene or making a protein that we want lots of that DNA. All right, so to work directly with specific genes, scientists prepare gene-sized pieces of DNA in identical copies. This process is called gene cloning. All right, so cloning is making identical copies of something, and so this is gene cloning because what are we making identical copies of? A segment of DNA, which is a gene. DNA cloning and its applications, a preview. Most methods for cloning pieces of DNA in the laboratory share general features such as the use of bacteria and their plasmids. So remember, just a quick little review, that bacteria have here's the bacteria, have the one big circular chromosome, and then they have extra circular pieces of DNA called plasmids. So these guys are the plasmids. So we're going to use plasmids and talk about those. Cloned genes are useful for making copies of a particular gene and producing a gene product. All right, so we're going to learn about how we make copies of it and how we get lots of the product that gene codes for. All right, so next you have this picture here. This picture here shows you some what we do in gene cloning. So let's look at this here. So let's say we want to clone a gene of interest. So notice here it says cell containing gene of interest. So this could be a human cell. And here's one of the chromosomes of this human cell. And so what we do is the gene is just part of that. Okay, so you have the gene of interest, or actually right here, the gene of interest. Let's say it's the gene that codes for making insulin, which is a protein that we need to help get glucose into our cells to do cellular respiration. So the gene of interest is the insulin gene. So that's the first step. The second step is we take a bacteria. Now remember, the bacteria have one big circular chromosome and they have those extra pieces of DNA, those circular plasmids. So we isolate and take out the plasmid. Then what we do is we cut open the plasmid. So here's the plasmid, purple here, to put together. We make one cut, and then insert that gene of interest. So right here is the gene of interest. So now we have a part bacterial, part human piece of DNA, little circular plasmid, because it now has a human gene, the gene of interest. Then we put that plasmid back into the bacteria. And so now this bacteria is a recombinant bacterium. Why? Because it has DNA from 2 different sources, bacterial and human. All right, so it's a recombinant that's been recombined. So then bacteria do what they do. So if we grow it on a medium, they start dividing, and basically, remember, they divide by binary fission, and you produce lots of clones of this bacteria. And so now you have a bunch of bacteria that have the gene of interest. All right, so it says host cell grown in culture to form a clone of cells containing the cloned gene of interest. And so then what happens is we can go one of 2 different pathways. So this notice here this arrow here, and then there's another arrow this way and an arrow this way. So what do? We use some of these bacteria, and we use them really just as cloning machines to clone lots of copies of the human DNA. So as this bacteria divides, they essentially make the copies of the human DNA. So then what we can do is isolate those copies. So then notice here it says copies of the gene. And so we can isolate those copies and do research on them. We can try and put those genes and put them into other organisms. So we've done this, for instance, making plants pest resistant, so pests will not want to eat them, and therefore it helps farmers and crops. We've used them by inserting these genes into bacteria for cleaning up toxic waste. So this picture is showing you an oil spill, so we can do research and so on, and by inserting these genes and practice doing that. On the flip side, on the right side of the diagram, it's showing you that we can actually use the bacteria and allow them to express the gene of interest. What does express mean? It means they're making the protein. So the protein is expressed, all right? So it's making this right here is the human, this right here would be human protein that it's making. And then we can isolate the protein, so we can harvest the protein. And so now we can do research on the proteins. So we can do research of proteins that dissolve blood clots in heart attack therapy. We've done this with human growth hormone that treats stunted growth, and so then people can have a mutation in their gene so they don't make enough human growth hormone can be given human growth hormone that originally came from a bacteria. So we put the human growth hormone gene into a plasmid, inserted it into the bacteria produces the human growth hormone, and you can give it that way. The gene for insulin does this as well. My husband is a type 1 diabetic. He has to take insulin with every meal so that his glucose in his meal can get into the cells of his body so his cells can do cellular respiration. And when you look at the packaging for the insulin and you read the fine print, a lot of people don't, but I did, and it actually tells you the cultures of the bacteria that were used to grow. So there's recombinant bacteria that have the human insulin gene that makes the insulin and so on. All right, so those are some applications for this. So now we're going to get to the nitty-gritty about how in the world do some of these things happen. So how do we cut the gene? How do we get cut the plasmid? How do we get the gene in, and so on and so forth? And so the bacteria has something called restriction enzymes. So using restriction enzymes to make recombinant DNA. Bacterial restriction enzymes cut DNA molecules at DNA sequences called restriction sites. A restriction enzyme usually makes many cuts yielding restriction fragments. So it cuts up the DNA and you get all these little fragments. The most useful restriction enzymes cut DNA in a staggered way, producing fragments with what we call sticky ends that bond with complementary sticky ends of other fragments. So we'll look at this in a picture in just a second. And then we take our good friend DNA ligase. Remember ligase from DNA replication? It's an enzyme that seals the bonds between DNA. Remember, it sealed the bonds between the Okazaki fragments? Well, now we're going to use it for something purposeful in DNA technology. So that's what I meant by we're going to take what we know and use it a little bit differently, manipulate it to do some things that we want to do as humans. So you have this picture here. So here you have your restriction site. So this is the restriction site is the cutting site for restriction enzymes. Okay, so notice here that the cutting site, the order of the bases on the top, if you read it from left to right, GAATTC. And then you look at the bottom and read it from right to left, GAATTC. It's the same order of bases, just in opposite order. So what happens is the restriction enzyme cuts between the G and the A of the sequence GAATTC. So it looks, this enzyme, these restriction enzymes, which bacteria have naturally, by the way. Restriction enzymes are in bacteria, because remember, bacteria can take up DNA, they can take up foreign DNA and so on. Restriction enzymes will cut up that foreign DNA as a part of protection measurements. So bacteria naturally have these restriction enzymes. So you have this restriction site. And so relating this back to our plasmid, so if we draw over here, remember you have your plasmid, that we want to cut open the plasmid so that we can put the gene of interest in. So let me get a different color here. So the DNA here is double-stranded, so somewhere on this plasmid is this cutting site, GAATTC. So then the other side is the exact same here, GAATTC. Okay, so so when we're looking at this picture here, this picture is just showing you a section of the plasmid. So what's going to happen is this enzyme, this restriction enzyme here, is going to come over here, and any time there's that cutting site, it's going to cut between the G and the A of that particular sequence. So it's going to cut here and here. All right? But remember that the A's and the T's here are only held together by hydrogen bonds. So basically those hydrogen bonds break. So I'm going to draw here and connect those. So when the restriction enzyme cuts this plasmid, it cuts it in a jagged cut like this because of the way that the cutting site is the same frontwards and backwards on the two respective sides of the DNA. And the purpose, we want to make sure when you pick a restriction enzyme, is that there's only one cutting site, so the plasmid only opens at one spot. So the plasmid, after this cuts, is going to look like this. All right, and so you're just going to have one cut, but the ends here, the end here and the here, are going to look like this. And so this part right here is really this part right here. So, so because of that jagged cut, so you're going to have here, it cut between the G and the A, so you have that cut. And then it did up here, the hydrogen bonds break, and it did the G, cut between the G and the A here. So now you have this GAATTC. This AATTC is no longer bonded to anything, and this AATT is no longer bonded to anything. And this is what we call a sticky end. Why? Because if this comes in contact with a TTAA, it'll bond back together. All right, and so that's why it's called sticky ends. And so now, remember, what do we want to do with this plasmid? We want to insert a human gene into it. So we want to insert the human gene. Let's do here. So now we take, here's the human DNA. So this is human. So here's your human DNA. And the human DNA has a gene. So this is your gene of interest. So let's say it's the human insulin gene. So then what we want to do is we want to cut this gene out of the big long piece of human DNA. We don't want to stick every single gene on this chromosome into this plasmid here. So basically what we're going to do is use that same restriction enzyme, and we're going to find using that same restriction enzyme that's going to have a cutting site, GAATTC, T-C, and then G-A-A-T-T-C on both sides. So it's going to have it on both sides, A-T-T-C, G-A-A-T-T-C. So it has it on both sides. And then what's going to happen is it's going to cut here, that restriction enzyme between the G and the A, and cut here between the G and the A. And so now you're going to have this fragment. This is called a restriction fragment that contains the gene of interest. But because we use the same restriction enzyme, they have the same sticky ends. So this part right here is this here. So when we cut this out here, this is what they're showing you here. Here's the human DNA. And when we cut it, it has the AATT over here, just like this. And it has the TTAA over here, just like that. So now, when we put this plasmid with this cutout gene, the sticky ends, notice here, the sticky ends of the plasmid, the TAA, are going to bond with the AATT here. And then the other end of the plasmid that has this sticky end is going to bond right here, and they're going to bond together. And so what does that look like here? Here's your plasmid. And then your gene of interest, I'll put in red. So now they're right here. They have hydrogen bonded together, the sticky ends. And so then the thing, though, is that we need to hook them together, kind of like the Okazaki fragments, and in comes the DNA ligase. The DNA ligase is going to glue them together, covalently bond them together. So now we have one plasmid that has all the bacteria genes, but now has the human gene of interest, which is your human insulin gene. And so it has that dark spot is supposed to be the gene in there. So it has that gene. And so this is what we would call, this is a recombinant DNA, which is what this does here. All right, so this is our recombinant plasmid. Okay? And that's how we make the recombinant plasmid. Then, as we talked about earlier, so that's the logistics of how this happened. This whole first step, that's all that occurred right here, is now we have a recombinant DNA plasmid. So then we have to mix it with the bacteria, and remember, bacteria naturally take in naked foreign DNA. So if you mix the plasmids with the bacteria, some of the bacteria will take up these plasmids and then have that human insulin gene in them and can either make the protein, or we can allow them to divide and make many copies of that.